Mexico, we are around 70% compliance. The goals, the main, not the only ones, to prevent, to reduce significantly ocean pollution. We can assure that is the main concern of everybody. How are we polluting coastal areas, the oceans, and how is the ocean responding to this threat? Minimize acidification of the oceans. Regulations about fishing, business, sources there. We have to ban some aspects of excessive fishing. Projects, current projects. That remain. We have string fishing and hook fishing and contamination by plastics. This is a global commitment for the economies to have clean oceans. We'll hear more about it. National plan of action about residues and plastic contamination. It's called REMAR. 9 of the most relevant agreements in terms of the actions that must be conducted to assure sustainability. As you can see, many of them because a great exploitation of extraction, transportation of oil. 4 of these have to do with these accidents, control, fuels. They have to do with contamination, not just by oils, but any other substance that is getting in contact with coastal areas. We have Mexico, different institutions that will help us to manage our oceans. Among them, these are the 4 main ones. Intersectorial, general directions from meteorological, interinstitutional collaboration for oceanic research, and national institution for research are made up of different stakeholders, but all of them are presided by Marine Secretariat, who organizes, generates these guidelines. Where do we want to do actions, programs? We have a law. It has been very complicated because every sector wants to get the benefit. So if you have environmental, social sector, productive sector in the same table, everybody wants to get the most. But we have reached an agreement with a certain level of governance to generate this national policy of oceans and coastal policies. What you can see here is who are leading this or implementing these policies of this document in Mexico. Through CIMARIS, we have all of these secretariats involved. Each one of them with its jurisdiction, limits, its own bureaucracy. So even if the paper is stated that things are clear, but the reality is not expedite, it's complicated, the articulation of all of the sectors. So what we're looking for with this national policy of oceans and coastlines to improve the livelihood of these inhabitants, which tend to be very complicated from the economic and educational point of view and so forth. Also, to have a sustainable use of natural resources. We want to strengthen local economies, improving competitiveness of all of those local economies, regionally speaking, and later, internationally, we want to ensure that ecosystems here are. This is what causes a little bit of conflict, because this instrument mentioned that they don't suffer irreversible alterations. Let's use electricity, but let's use it the best way possible, but let's not abuse it. So, from a point of view, romantic, poetic point of view, gets to the heart, but it's a reality, it's a commitment between the use of the resource and the benefit for the human being. I want to talk about the interinstitutional efforts. I just mentioned 3 examples that have provided the results, that have a positive impact. The last one is being developed, is the one I would like to address. These 3 efforts, the first one, CIMAR, and then we have HydroGuard. CICOM is a consortium of research in the Gulf of Mexico. It came up in 2015 for researching and consulting services. Why in the Gulf of Mexico? As you are aware of, in this area we have abundant hydrofuels. What was the baseline? We didn't know. In every matrix present over there, water, all levels, every level, organisms, waters, at every level. So this consortium came up to fill this void of information for Mexico. Was implemented this project, this implementation of networks of observations, ecological and so forth, 3 years of campaigns throughout Gulf of Mexico, so you can imagine huge number of data that was gathered. All of this was analyzed. As I mentioned, we have to deliver scientific, the PhD, any value, we deal both, but we have to deliver this information to be useful for society and the decision makers. So we developed this atlas. So you can download them, they're free of charge. Atlas that goes from biochemical aspects of risk, turtles, so forth. Hydrofuels is the one I had the pleasure to be a co-author. So I invite you to download these documents, which are very useful. Some of the things that we can observe in hydrofuels, the whole Gulf of Mexico is an important effort. The other system that we have is information and analysis of coastal area. Was developed Conario in Mexico. It's a data center that seeks to generate early warnings and analysis What you have here is La Liga. It's an open system, but it's public. I invite you to visit. What you will find, the integrations of big data, big volumes of data that are analyzed, that are managed, visualized to generate tools that could support the decision-making process and the information for Mexico society, early warnings in the case of meteors, for example, and we analyze timing, biological and physical, chemical variation to see what is going on in the Gulf. This is the website. What you will see, all of this information they manage, if some of the information is based in data taken on-site, hard data, most of them are through satellite. We're working in the process of validating this information. So everything that you observe, nitrate, amount of concentrations, this has to do with real data to determining if this can be validated having about of water, and you can have information about if it is correct or not. These are the variables that are being measured. This type of figures, you can see them. You can interact in the platform with all of them. What date you want to see, what type of parameter, the same way. We have strengthened this platform with all the information of all the organizations that provide you information of the population, the use of soil, all the physical, economical variables interact with this platform to generate scenarios, models that could explain causalities, predictive models to be developed. This, for example, about the dispersion of caffeine index. Of course, the fish they don't drink caffeine, so we can see what's in the lagoon. This I cross it over with the incidence of pathogen organisms or any other variable in terms of human terms. Gastrointestinal issues, can start reading this process to identify the problem, to generate a solution that would be really efficient. And as finally, I think I have 1 minute, 2 minutes. Okay. Finally, what is relevant right now for the future of the oceans and the water? General terms, not just in Mexico and internationally, is to have management systems to allow us to understand resources we have and to manage them efficiently and to manage them correctly. What do we require? We can have these efforts, but if we connect them with your own efforts or the rest of the efforts of the community, north, south, can you imagine the results, the efficiency of managing we will have of the resources? It will be incredible. Mexico, it's on its way to compliance with all the CISOS. So keep working. Have a lot of work to do scientifically as communities, with business, and focusing on the water and environment. All of our approach is being modified. I just want to thank you to the National University of Costa Rica, to all of the sponsors, and all of those who are involved in this. We thank you from the heart, and I want to thank volunteers, organizers, colleagues that have made this event and these systems possible, and to develop this management system for the underwater sources. We have theses at every level that you can check, you can research, you can tell your students. If you have not sent notice, you begin an investigation. We invite you to consult our networks and hope you can participate with us. Thank you. Thank you, Dr. Arcega, for the presentation. Next, Dr. Rafael González Quiroz, Director, Oceanography Center Beyond, one of the Spanish oceanographic labs, and he was former Director of Investigation and Ocean Research Center. Represent Spain in the UNESCO, and he works for Oceans of the United Nations. Dr. González, observation, disciplinary of the ocean in Spain. Thank you for the volunteers and all of you present. I'll talk about a vision, general vision, partial vision, of course, about biological observation, physical oceanographic, use of the Spanish Institute. It's one of the research centers that specifically deals with observation, systematic conservation. Our institution was founded more than 100 years in 1814. Right now we are about 850 persons working in it. We have headquarters in Madrid. We have a direct relation, very fluid relation with several ministries who have competences in the marine environment, and we have coastal centers around the country. We have integrated
en el Consejo Superior de Investigaciones Científicas, que es el organismo mayor que hay en España dedicado a la ciencia. Somos actualmente 14.000 personas y dentro del CSIC hay varios laboratorios marinos, con lo cual ahora nos hemos integrado y creo que podemos además salir reforzados en los dos sentidos, porque teníamos distinta orientación.
We had a different approach, but now we are complementary in many areas. We have other laboratories which depended from autonomous communities about the coastal resources. They have research centers, observation centers, and besides that we have all the universities. It's difficult to have the total number of researchers in terms of ocean sciences. This is the structure, chart flow. We have our directorate and two areas, advising technical area. One of our main mandates is to advise the state about sciences in order to manage things in the ocean, and the scientific research and advice. We have coastal centers, we have researchers working, the central office in Madrid. Every investigator is under this umbrella, but depending on the activity at a certain point we channel. And with the number of investigators, some of them work what they do is just research, others advise. We try to have a balance between these two things, and this is what we do, to make science, but directly to advise the state. Resources, oceanographic fleet, circle, those who have Spanish Institute of Oceanography. The biggest one are part of the FICID, where we are integrated, and we're trying to manage all of this fleet. And we have some other vessels from the Secretariat of Fisheries, and we use oceanographic researchers to conduct different tasks and to advise management fishing resources. And this will be my talk, observation, systematic observation of the oceans. Systematic meaning is not part of the projects of research. It's done on temporal scales and special scales that exceed what normally a research project of 3 or 4 years do. And big part of our budget is related to managing, to provide the state with the scientific information for final management. This allows us to dedicate part of these resources, as I will explain, to begin temporal series of oceanographic information that we thought from the point of view cannot be solved with just research projects like long research projects or very ample areas. So we need to conduct mappings to be part of the project. This is fundamental to respond to scientific issues, the distribution, scale, patterns of all systems, to deal with questions of variability in long term of ecosystems that is related with climate change and its effects, and especially this series. These time series are fundamental to provide a scientific advice to the states, protected marine areas, planning, plastics contamination.
All this information will not come out from the research project, but directly from making observations, as I mentioned, are systematic. Examples of this, well, as I said before, well, fisheries, conservation, things that have to do with to facilitate the marine spatial information or technologies that depend a lot on the state. From my point of view, in the legal frameworks that are associated to the management of these resources, it is fundamental so that we might see these type of observations. If not, city that has attached a 30-30 objective in which we are all of the countries trying to work so as to have 30% of the surface ocean under some sort of a protection figure. Another one is the framework of the marine strategies. Well, it tries to reach a good environmental state of the European seas, oceans, so that this is something that we might be discussing as to what is a good or bad environmental state. But the fundamental thing is that, as we mentioned, it puts into operation a series of programs for monitoring, or invites the members to do so that allow us to determine what is the ecosystem. So, therefore, having that type of systematic information. This allows marine spatial planning and so forth. And finally, the methodological predictions. Every time, especially with all these models for the predictions at the midterm, 10 days a week, we know that they have a very high dependence on the oceanic data. The science of the new said that if you want to thank somebody for tomorrow's methodological prediction, you can thank a methodologist, but if you want to predict 1 week from now, you have to thank an oceanographer for that. because of the role that the ocean plays in the regulation. As we said, all this tradition of advisory, especially in the fisheries in the 1980s, this transformed the institution so that we could provide this scientific advisory towards the fishery, even going beyond, taking it far further and beyond to do science. This was something that was not seen very nicely within the institution. Unfortunately, this has changed a lot. We have no traces left of that mentality, but with that function of advisory. And that's what we do, campaigns for our coast, with acoustic, with fisheries, experimental and so forth, to determine what is the state of the stocks, and based on that, to have maps of distribution that later can be used also to make science, to relate with the temperature of the water and things like the ones that I'm putting there. But somehow the systematic information began with the fishery management, and within these campaigns we began, apart from extracting the information directly that is related to the fishery information, we started taking out all the different analyses that were made to look at the two funds, etc. So with this, we are coming to an agro-systemic approach on fisheries, so that you might have an idea of the magnitude and the effort that this presupposes. These are the data of recapture and value thousands of euros from the European fleet in 8 months, and this requires to collect information regarding the capture, analyze it, and provide the states of advisory for the operations in all these different types of zones. As a matter of fact, the European Union even demands that if the Spanish fleet is exploring resources in an African country, well, we have to make the studies to see if it is done in a sustainable manner. And many times, the Spanish fleet does not look at what happens when they go fishing in the coast of these countries. As I said before, those funds associated to the various three systematic exploitation along some of the points of the coast, and here we have represented all the stations where we make exploitation. We collect information on a repeated manner. This is something that began in the year 1987 in La Coruña, in the northwest of the Iberic Peninsula. And since then, in all the northern zone, all those red points that you see there, we do a collection of data on a monthly basis so that we might have the information of the temperature of the water, the nutrients, and the plants. And this program was expanding zone by zone to different zones, in which it is done with different frequencies. And well, we also have represented the network of the seas. That is something that began much before even the Spanish Civil War, and now it is being complemented with buoys and other type of surveys or studies. And as it was being commented, these are the series of 3 Spanish information with uninterrupted data. That we even have some data from 1920, but they are still maintained, and you can see the trend of the increase of the level of the sea in all of them. And this is an example of that coastal station that I was mentioning from La Coruña, which was the first one, starting 2012. And as you saw in the frame, work right in front of the lab, we have a weekly sampling. And this graph is comparing the results that you obtain if you sample monthly or if you sample on a monthly basis. Weekly it gives a GTD with a probe of salinity, and you can also get the data of Profila. Also, but this one has a lot of more parameters, but this is a coastal zone that is very dynamic, and it gives us an idea of what we're missing out on when we don't do it. At the end, we have to put a limit somewhere. So this is another probe that I'm telling you about these stations with occasionals of the zooplankton, where we did analysis to see if there was a change in the amplitude of the fluctuations. And we see that after a year, we see that there's an increase in the biomass, and we have come to the conclusion that this is related to environmental parameters of flourishing and others. And more recently, Spain has made a long contribution to the CISOS system of buoys that are throughout the world, and that their contributions in the world are accessible for anybody. And these system that is collecting the information of the first 2 meters of the water layer and is advancing towards the 4,000. And this has allowed us to determine that the ocean has absorbed more than 90% of the excessive heat that we have developed with greenhouse effect gases. Before we thought that it was close to 30, but we have seen that now we're short of that number. Another thing that we have seen with the observation, establishing the protected area, protected marine areas, and this is something we are financing with the investigation, with the research that has allowed us to advance and to see what zones we want to protect and what measurements we have to protect them. This scientific information, and currently Spain has a protection of 22%, and we hope to reach 25% by the next year, walking very nicely towards the objective of the 2030 goal, sorry. And this is based on non-invasive invasive systems that have, for example, this is done to make a tri-dimensional reconstruction to analyze the abundance in some of the cases through the management of artificial, and based on that we can generate models of distribution that will allow us to see where my conservation has to be taken. And in the video we are seeing especially in the profound zones that require the use of oceanographic vessels and so forth. And as I said before, to reinforce all this action of monitoring of the marine strategy, as it is known in Europe, it has a series of descriptions that cover basically the totality of the matters related to the ecosystems, the marine ecosystems, and that need to put in monitoring programs in situ. This has been very difficult because of problems of all types, financial, bureaucratic, and others. But I think that this has finally taken off very strongly in Spain, and I think that this is a moment in which we can have a system that is very decent for the observation of all the parameters related to the systems and the generation of indicators also. And here finally, talking about when things go wrong, the research projects, and when we come to the management of natural disasters. This is a lagoon, a coastal lagoon that we have in Spain, in the region of Murcia, that because of the increase of the tourism and especially of the agriculture, in view of the lagoon, it has generated a ultra-oxidation effect that is very strong. And if we had some temporary observations of some months, we could have foreseen that this was going to happen, but we didn't. So now we're working along with a group of organisms to try to put an end to that. And before I was talking about the observation of physical and biochemical observation that is not associated directly to a mandate, but that is fundamental for providing the base of knowledge for the management of all the ecosystems. And then we have the relationships that we have through methodology and so forth. In Spain, we don't have a mandate of who has to be in charge of that. And we have been more or less covering this among a group of organisms of ports of the state that have a system of buoys, surfaces that is automated, that is very well built, and the network, as I was mentioning before, 2 infrastructures that also participate in some of the programs for observation. Some universities and that have put together this committee, and this was a little bit of the initiative of the IEO, but out of the initiative of all of us, but it doesn't have a direct support or initialization of the government. So it's not built with the structure of the state, and that is what we're trying. We're trying to work on that. We're trying to convince the politicians that it is necessary to coordinate all the observation we do in Spain and to do it in a coordinated manner without having to resign each one of us to what we do. That's it on my part. Thank you very much. And you have right there my contact information in case somebody wants to make any question later on. And we can go on to something else right now. Thank you very much. Thank you very much to Dr. Gonzalez. We hope that some of those vessels that you showed us might visit us soon. Or the one from Mexico that is even closer and very modern, as I was being told. Okay, and following, we are going to have the presentation by Dr. Giuseppe Sualia. He is an investigator, a researcher of the School of Science and Marine Sciences from Italy. He has participated in more than 30 oceanographic expeditions. In 2027, he participated in the exposition of the circumdating the Antarctic as responsible of microplastics. He participates in several international and national expeditions, and he has more than 90 published articles. Dr. Suarez is going to present to us monitoring of the microplastic in marine environments in the perspective. Mientras tanto, muchas gracias. I'm sorry about my English, but I'm going to talk in English. Please forgive us for the technical inconvenience. We are going to go on to the next presentation. Let's see what we're told. Okay, so Dr. Josep Gaston Piqué, he is a researcher for the Institute of the Sciences of the Oceans from the Scientific Experimentation of Spain. He has more than 300 experimentations jobs published. He has directed oceanographic research, and the topic of this presentation is microbes as sentinels of the planetary health. Well, thank you very much for still being here, and that we are not all asleep. And What I'm going to do in this presentation is to defend what the people do when they come into a book reading. Well, I come here to defend my book because I'm going to talk to you about the microbes, of how they work at Sentinels, and this is an idea of how the microsystems works. So the first thing that we all know, but sometimes we forget, is that the majority of the biosphere, most of the live beings, is, by the way, microorganisms. This is a distribution of the live beings on Earth, and evidently we know that there are a lot of plants, but when the plants are not there, it is the animals who are very protected, and humans is another part, but the only thing that we do is destroy the planet. But when we access, we are greater when we see the sea. This is a distribution of the biomass of the planet Earth. In this case, of the planet ocean. There are many, but apart from that, they grow very quickly because they're small. The allometry that everybody knows of the size versus the rate of growth, it makes that the microorganisms, they can come and divide up to maybe 10, 20, 30 times per day. So therefore, what's going to happen is that they're going to have also a capacity for evolution that is very quick. This graph, according to maybe that's something that sounded to many of you, because during a year and a half or two, we were suffering the topic related to COVID, and this is the evolution of the COVID virus between May 2020 and August 2021. There were viruses generated by very different from the previous ones during years. Those evolutionary changes also happen. This is an experiment that is very much fun, that is made by the Richard Lance, the investigator in the United States, who, I can't read it, but I think that it was for 25 years he dedicated to talk about E. coli, to maintain it, and he passed it on from one Petri plate to another Petri plate, and every now and then they kept a copy of the strains for the later molecular study. And one of the things that they saw is that in this experiment of 50,000 generations, there had been, for example, a reduction in the cells. The cells had a smaller genome, as we can see it in genome, as we can see it on the top, and the number of mutations had increased throughout time. So therefore, in the microorganisms, we have adaptation that is sufficiently fast, and this doesn't work. Where do I have to write it down? Okay, sorry. Now, go back, okay. This adaptation at the same time is very important because the microorganisms are the engine that moves all the biochemicals in the ocean. I haven't done anything. Should I click on it again? I don't know. I could tell some jokes, but I don't know if you're going to enjoy it. I don't think I'm that good telling jokes. All the biochemicals of the transformation of the compounds, the fixation of carbon, the photosynthesis, production of methane, the consumption of methane, the nitrogen in the ocean are a fraction that is enormous or dominated by the organisms. It is relevant in terms of biochemical of the oceans, to the point in which we are capable of seeing their presence, and they're actually acting in the ocean from the space, like this bloom of floral cocoons in the coast of Florida. And they also respond very quickly to the environmental change. This is an experiment that we carried out in which we changed typification. We changed the sample of water, and we saw it for 10 days, and we saw genetic changes that were very important in the expression of the genes in those samples of water, for the simple fact of just changing the acidification of the water. Apart from that, in this concrete case, this depended on the chlorophyll that the sample had, that sometimes was not. When it's the lowest, it was more sensitive to the system, to the acidification. Or for example, these observations that in 4 or 5 4, 5, or 6 years. There we can see that it's a study that was carried out by the UN, one of those temporary studies that was mentioned by the previous presenter, and we have seen that it changed the size of the batteries, or that in the case, it changed the team of algae, the type of algae, and it changed for 4 years. So additionally, it allows me the bacteria allows us to see that many times regarding the ecosystem. This is a study that was carried out by Carolina Martin, here present, in the Gulf of Nicoya, in which what we can see in the CISOS study, in one of the studies, how there was a present, and you can see the brown bars with a round circle in the Gulf of Nicoya. Luckily, this happened only during the dry season. When the rainy season came, that disappeared. But this means that the microorganisms are telling us how the ecosystem is. And therefore, don't everybody forget that the microorganisms are very abundant, and they grow very fast, and they evolve. They evolve very quickly. They affect the recycling or the geochemical cycles, and they have a reaction to the change of the ecosystem that is very quick, and they can convert, and they become indicators. So sometimes we say that microbes, and this is something that we saw many times from our colleagues in medicine, that they say, no, that your intestinal flora is telling us, flora is telling us how your health is. Well, we can say that the microorganisms are also the sentinels of the health of the ecosystem, or sentinels of the health of the ocean, in this case, or of the planet, because the ocean is a majority of the planet, as we all know it perfectly. So in spite of the fact that we all like to think that the ocean is like that, in spite of the fact that we think that this is what we are interested in, because that's how we attract the tourists, the ocean is like this. The ocean is dominated by microorganisms that respond more quickly, that tell us what is happening, or that something is happening before the turtles tell us. So therefore, it is very important to use the microorganisms as the indicators, as the sentinels for the change that the ocean is suffering. In this case, in addition, the microorganisms are extremely important for the biotechnology. In this presentation, I had it longer. I had put examples of compounds. I had put an example of the compounds of polysaccharides, antibiotics, of biosynthetics, of tools for molecular addition to have systems that we're finding in the microorganisms of the ocean, or of biogeneration of mercury, for example. I'm just going to take one. And why this is important? Because this let us know a little bit about it all. This is a little bit of the scientific biomass in the oceans. Our colleagues from Teleoceans collected all the molecular data that they had. They constructed artificial genomes, in the sense that they are genomes based on metagenomes, and they look for synthetic imposition. What they found is that in some of the samples that you see, that are the ones that you see in black on the left side, it's something that we cannot, it's a problem that we cannot point here. There was a great number of genes that were significantly active. They are building some of the molecules of which we know nothing, and the organisms we didn't even know the name that they had, and now I don't know what name they have, and I don't see it because it is way too far, and I don't have my eyesight that's sufficiently good. But the microbiomes, we put microsplenea for oceans, another one, because they were obtained in these species. So, apart from that, these are my favorite. That is more than 5% of the communities, microbial communities in the bottom of the sea of the Atlantic and the Pacific. So, I've been asking myself in Spain if we had the figure of the sereno, the watchman, the one night person, and they say that in many countries they had also existed. The sereno, there was a person who had all the keys of all the houses, and that would see, and he was available in the house, and people would call him up and tell him, hey, open my door. He was a person who knew how to come into all the houses. He knew what there was in each one of the houses, but he wouldn't say it. These are the microorganisms. They are the ones that open the doors to multiple actions, to multiple things, and definitely we have to interrogate them. How will we do this? Up to now, what we have done is a job that is spatial, jobs like the Tara Oceans, in which a ship would go around the world collecting samples and sequencing the genome of all the different samples of metagenomes of all the samples. This is what we call a 3D exploration, because the space and the depth. It takes into consideration the space and the depth. And here we have what was collected by the Tara Ocean, the Malespina, Spain, from between the year 2010 and 2014. And just to give you an idea, the Tara Oceans in the first analysis was able to describe 40 million microbial genes that are not redundant, that are different. In the second interaction, they found 47 million. The Malespina, we are a little more discreet. In the first round, in the first analysis, we came to 1.2 million non-redundant genes, and in the second analysis to 17 million non-redundant genes. Nobody can analyze all this. Nobody can know how much useful things there might be hidden in this amount of genes that we're talking about. So I think that we have to advance to this tri-dimensional and quadridimensional genome, which is what we were being talked about in the previous presentation. And we have to go and establish observatories, permanent observatories, in which we can analyze the presence of microorganisms and the genome changes of those microorganisms. So we have this map. It's a little old. It's from 2017. But if you look, there are no microbial observatories in the tropical zone. We have none. In Central America, there is practically nothing. Now there is one. But there was in Australia. It was a job that we are about to finish, in which we are comparing several microbial observatories that we are analyzing on the long term. And what we're seeing is that the sea is very little studied in terms of the changes that are.
These seasonal changes can be interesting. This is a figure. Branis is from north of Barcelona since 2004-2014, changes in the bacterial changes. And it varies from one year to another, but not much. There was a period where we had huge changes. We had peaks, different colors. These coincide with destruction of a port. They were providing information that we would not be able to see. They were building a port, not destroying the port. For temperature also we are seeing changes, rates in red, annual, different group of diatomeas, algae. There is an annual change. Our ocean is becoming more endotrophic. This affects fisheries. We can see the genomes changes. We look for mutations, comparing different genomes in different times. Mutations, synonymous, non-synonymous, we evaluate. We have been able to see, like in a place in France, which is similar, where we have analyzed 7 years, 27 bacteria organisms that we have found that are changing. We don't know where are they heading, but we can mark the change. What do we need to do to change? We are analyzing things when we can. Sampling is very expensive. vessels are not available for everyone all the time, so it's difficult to have a consistent, regular campaign. So what we have to do is to try to make these studies. Every day bacteria are changing. We cannot sample them every 2 months. We are losing what is happening in those 2 months. In this figure, 50 days, 40 days, if you see the colors up there, we seem to be the same. Green, but similar changes in the lower figure. Those peaks. It's an organism that appears for 1 or 2 days and disappears. So we need to see what's the dynamic behavior. We need technology, not just sampling capability, but technology. These are flow cytometers that measure, count cells of the plankton and classify them according to its characteristics, and they do it automatically. We have placed one in an oceanographic vessel, but this allows us to measure every 15 minutes the concentration of microorganisms and its diversities. For example, in this campaign, in this ship that would just retire, we are seeing, for example, how moving from the center of the ocean between Catalonia and the Balear Islands, we had changes. We see changes in the diversity of the communities. We had areas with a higher density, we don't know why, and others with lower density, we don't know why. Finally, what we have to do is to relate the presence of organisms to pollution. We have those genomes, they're doing something. We have to use every day RMA if it's being produced more in a different time of the day. We have a lot of work to analyze, a lot of genes, you don't have to see them, but throughout the day, what we measure is at what time of the day each organism is changing its function. We do it with this devices, environmental sample processors. It's very difficult for them to work, but we have to go this way in order to have the proper bioinformatic knowledge, to listen, to see constantly. This is organisms that are abundant. They grow a lot, they evolve very fast, they react to the changes immediately. Very good indicators about the health of the ecosystem in our relevant in cycling, chemical processes, and there are sources. Bioecological products. I have to plea for, if you have money, we want long-term monitoring in sight. Every country should have 4 or 5 sites, very well monitored, long-term, not to manage them 1 year, 2 years, but 10, 15, 20. Shorter spatial and temporal scales, coupling, omics with biochemical rate measurements, developing technologies. And as I mentioned in my book, thank you for your attention. We try again. Gracias por la invitación. Hicimos investigación en todos los temas, especialmente en el Instituto de Ambiente Marino. Este simposio es sobre monitoreo biológico y comencé a hacer mi investigación recolectando muestras de la región. Estuve trabajando en un proyecto grande europeo para estudiar en distintas etapas cómo se movilizaban en las corrientes en el Mediterráneo. Y comencé recolectando muestras de la capa superficial del mar. La situación me fue clara de que mis muestras estaban llenas de miles de fragmentos de plástico, estos fragmentos más abundantes que los organismos que estábamos investigando en el proyecto. Decidimos cambiar mi motivo de investigación hacia las partículas de plásticas. Extraíamos más de 1.400 fragmentos. Hicimos un análisis químico y estimamos cuántos microplásticos flotaban en el mar Mediterráneo. Al mismo tiempo, durante la navegación, diferentes estaciones, hice monitoreo visual para los macroplásticos, viendo la superficie del mar a medida que navegábamos y usando diferentes métodos de monitoreo. Este trabajo se publicó en el 2014 y encontramos este montón. Sigue haciendo esto los últimos 10 años. Tenemos una base de datos, 10.000 ítems. Las tendencias interesantes no se han publicado, pero lo que pueden ver, los ítems se vuelven cada vez más pequeños. Y este fue en el 2013. Estos gráficos muestran el crecimiento exponencial sobre la contaminación de plásticos en años recientes, hace los últimos 2 o 3 años, son mucho más. Tenemos un crecimiento exponencial sobre el estudio de plásticos. Está disparando y sigue la tendencia global de la producción de plásticos, que sigue la misma curva. Entonces me metí en esta lavadora. Comenzamos a recopilar muestras, compilándolas, mostrando y publicando, haciendo estimados globales sobre qué estaba flotando en la superficie. Y lo que ven, 4 ejemplos para las compilaciones más grandes de la distribución global de microplásticos. Las primeras en el 2014 y luego las actualizaciones en el 2021, 2022. Las compilaciones se le vio el mar Mediterráneo era el que más concentraciones tenía. Estos estimados coincidían con los estimados que hicimos. ¿Cuál es el problema? Si comparamos el estimado de la cantidad total de plásticos que se arroja al mar a nivel de escala mundial, esto es 8 millones de toneladas por año. Y este es el mismo orden de magnitud de todos los estimados que venía de los ríos principales, 3 millones de toneladas de plásticos. Y si comparábamos esto con todos los estimados globales de plástico que flota en el mar, vimos que había coincidía, porque estos estimados están en el orden de cientos de toneladas. ¿A dónde van todas esas toneladas? Una vez se producen. Entonces es un dilema del faltante plástico. Y ahora se está publicando mucho sobre esto. ¿Qué está pasando con el plástico que se produce? La forma de mostrar este concepto. Queríamos cerrar este ciclo plástico en el ecosistema. Para hacer, necesitamos cifras. el ingreso, cuánto plástico se arroja, cuánto plástico está en las líneas costeras o en el fondo marino. Estamos monitoreando para responder todo esto. Y lo que hicimos con carbón, aprendimos esta mañana. El CO2 y el ciclo de carbón es más estable y ahora tenemos una buena comprensión del reciclaje y el ciclo del carbón en el ambiente. Debemos hacer lo mismo. Lo interesante, la comunidad de carbón publicaba lo mismo que la comunidad de plásticos. Ahora está publicando. Así como se dijo en el 2019 que hacían falta depósitos de carbón. Trabajamos en un gran proyecto donde tratamos de desarrollar un sensor de plásticos, una meta ambiciosa, y luego sensores de microplásticos. Todavía continúa este proyecto. Tecnologías innovadoras, cámaras en drones o en aviones o barcos para monitorear la concentración de los plásticos. Y este programa de monitoreo puede llenar las brechas faltantes en el conocimiento. Satélites es una herramienta más poderosa que tenemos. No es fácil porque la resolución del satélite convencional no es suficientemente alta para detectar micro. Tenemos un documento donde podemos usar reemplazos y podemos detectar plásticos del espacio utilizando estas formas que reemplazan las tomas originales de los satélites. Sobre las brechas de conocimiento, viendo esta compilación global que les mostré antes, una de las cosas que me llamó la atención, este es un trabajo del 2013, pero aún en el 2023 no teníamos datos de Antártica ni del sur. La idea fue hacer una expedición histórica, por cierto, una expedición de navegación por Instituto Sueco, Instituto Polar Sueco, tuve la oportunidad de unirme a ellos, muestrear macroplásticos en el Mar del Sur, en el Océano Sur, muy pocos, 53 a través de todo el Océano Sur. La concentración era más alta al norte, arriba de los 45 grados. Recopilamos algunas muestras por microplásticos y tuvimos lo mismo. Extrajimos solo 7 microplásticos. Estas cifras son muy bajas. Las comparamos con lo que encontramos en el mar Atlántico, Océano Atlántico. Al regreso de Europa, la concentración es 10 veces más que la que vimos en Antártica. Si comparamos los que les mostré sobre el mar Mediterráneo, es 5.000 veces menos en Antártica. Estamos seguros que estábamos en el océano más limpio. Lo que yo veía en el Mediterráneo no se veía en otras partes del mundo. Y en Antártica era diferente hasta que nos dimos cuenta. Esa es una buena colaboración científica. Algunos colegas fitoplancton estaban filtrando agua de mar y seguían viendo estos colores, estas fibras. Nos las mostraron. Esto es parte de microplásticos. Vamos a mostrar esto. No estábamos seguros si estábamos muy cerca de la lavandería del barco. Tal vez eran muestras de la misma contaminación que estamos produciendo nosotros. Para evitar eso, volvimos a lo básico. Tomamos un balde, recolectamos agua en la superficie, la filtramos inmediatamente en el laboratorio y esto fue lo que encontramos, miles y miles de estas fibras textiles. Decidimos seguir muestreando en otras cuencas alrededor del mundo y de caracterizar globalmente sobre los contaminantes. Al final, contamos más de 23.000 fibras. La concentración que estaba entre... La concentración de fibras no es homogénea. El mar Mediterráneo, la concentración de algas no coincidían, pero la concentración de algas estaba en Antártida, no en el mar Índico ni en otros mares. Por alguna razón, estas fibras se acumularon en Antártica, lejos de los centros de población humana, pero lo interesante es que después de analizar casi 2.000 de estas fibras, nos dimos cuenta de que no son microplásticos, son fibras celulosas como algodón, porque abundan, no sabíamos, pero no esperábamos, solo el 80% eran sintéticas. La composición de estas fibras estaba con distintas bases oceánicas. Tratamos de hacer algunos estimados a nivel global, cuántas fibras están flotando en el mar. Las mismas, leímos los mismos órdenes de magnitud, eran miles de toneladas. La mayoría de estas fibras están por todo lado. Miles de trabajos se han publicado sobre estos, sobre las muestras. La mayoría no son sintéticas, son de celulosa. Y esto es una pregunta intrigante porque para la contaminación plástica tradicional, los polímeros más comunes son estos. Para textiles, la tendencia es lo otro. Producimos casi el 60% sintético para los textiles, pero ahora sintético es solo el 8%. No sabemos por qué lo sintético era solo el 8%. Las fibras de algodón liberan más fibras que las sintéticas. En el pasado estamos usando fibras de algodón, la mayoría, parte de la historia, y se han acumulado en el ambiente y probablemente no se degradan tan rápido como quisiéramos por los químicos y tinturas que se usaban en el proceso. Piensen en esto. Si queremos nuestras fibras, piensen en las banderas. Mientras estas son desprendimientos constantes, fibras de textil que se liberan al ambiente. La mayoría de estas fibras no solamente flotan en el aire, por eso en las regiones polares se encuentran, porque vienen del aire. En la literatura, la evidencia sobre esto. Ya se han encontrado estas fibras de algodón y sintéticas en los pulmones humanos. La liberación de estas fibras ha sido demostrado por varios documentos que lavar una prenda de algodón libera más fibras que las sintéticas. Y simplemente vistiendo un atuendo, una pieza, libera muchas fibras. Entonces, más importante, todas estas fibras liberadas en la atmósfera que las liberadas en las aguas negras. Por eso comenzamos a medir las infraestructuras marinas, con detectores precisos atmosféricos alrededor del mundo, y hay trabajos importantes. Este habla de una camiseta de un pescador después de 2 o 3 años. No tenía señal de degradación. El algo nos está degradando lo rápido que pensábamos. Por eso comenzamos a hacer algunos experimentos de degradación de agua profunda. Ahora tratamos de comprender más sobre qué le sucede a estos materiales en un ambiente como estos. Estas fibras en agua marina, las fibras plásticas tienden a flotar, estas otras tienden a hundirse. Entonces, encontramos concentraciones por debajo de la superficie, miles de metros por debajo en el mar Mediterráneo. También estamos estudiando estas hebras, el hundimiento en las estaciones en el Ártico. Estamos estableciendo trampas para comprender qué causa esta estacionalidad de la aparición de menos fibras. Terminé, solo veamos algunas transparencias, que es un problema grande. Se dijo antes, estas biomasas, si comparamos la biomasa de los animales vivos del planeta, la biomasa del plástico que producen por estos medios, estos polímeros, hace 60 años, ya es el doble de la biomasa animal. El plástico producido gigatoneladas, 8 gigatoneladas, mucho mayor que toda la biomasa combinada de los animales. El programa es grande, está frente a nosotros. Pensamos en limpiar. Las técnicas son útiles, pero no hay que olvidar, como aprendimos esta mañana, el profesor Gattuso, no podemos limpiar el piso si no cerramos la manguera. No se están degradando en el mar marino, en el ambiente marino. Falta mucho tiempo y de nuevo estamos usando soluciones naturales, piezas naturales, y ahora nos damos cuenta que liberan muchas más fibras en las lavadoras y estamos respirando más, si más se liberan. Esta es una transparencia fea, pero optimista. Muestra la historia de los contaminantes desde el DDT, lo que hemos resuelto, historias de éxito. Se predija que todos los contaminantes que se han identificado, hay una actividad que se está publicando, acciones regulatorias, y hay una tendencia constante con relación a estos contaminantes. Para microplásticos, las publicaciones científicas en el 2022. Tal vez estemos llegando a la publicación, al pico de publicaciones científicas y acciones regulatorias. Y esto está, hay negociaciones en términos de los tratados intergubernamentales del plástico, y esto es continuo, las negociaciones son continuas, nuestras esperanzas están ahí, no sabemos qué tan ambicioso va a ser este tratado, probablemente no mucho, según hemos aprendido otras cosas. Eso es todo desde mi punto de vista, lo siento por la tardanza, gracias por su atención. Thank you, Dr. Sonia, for his presentation. We'll take a 15-minute break so you can have a cup of coffee, stretch. Let's be back 5 minutes after 3.
All-in-one productivity software where you can manage everything and anything related to your work, your documents.
to the colleagues, we have put together an interesting group for different aspects related to Luciano, to the ocean and the sea. I would like to make a clarification before I go into the conversation in full. And maybe the presentation is going to be somewhat technical, but I think that it's necessary and very, very necessary because, for example, we all know that what resilience means. But the question is, how much can we identify if we use resilience as a quantitative indicator for the decision making? This is somewhat complicated. It's not very new. The other part is the part of exploited populations, which we have extracted the biomass. I'm going to refer very much to this, to the fishery. The fishery is the only activity of the primary sector that we don't have any input for the profession. Aquaculture and forestry, there are inputs to increase production. In the sea and the fishery, we depend on the amount of natural biomass production from the populations. And the other point is that if we identify the production capacity of a biomass, well, this changes in the current time with the climate change. When we talk about sustainability, we almost think about something. Almost always think about something stable. And when we talk about life systems, we're talking about a dynamic sustainability. And when we talk about biological production, this biological production changes depending on the production. So the presentation is going to talk about how that quantitative indicator works for the decision making in the case of fisheries at the level of ecosystems. Before it all, I would like to say a point in the part of how we conceive the fishery in these terms, the production. In nature, we have three great systems divided by population ecosystems. The individuals, we can measure the performance by the level of robustness. Be this that this represents more or less a biomass that put together the body, or the pressure of the metabolism. If the conditions are favorable, the organism develops favorably, and it can be successful. So the individuals are the populations, but the indicators of performance is not the same as the individuals. Let's say that the performance of the population is developed according to the persistence. If we have that the population, in spite of the fact that it is exploited, can maintain its persistence, well, we are doing a reasonable fishery, a healthy fishery. And the populations are part of the ecosystems. The ecosystems are identified by relations between the populations. The trophic interactions has a population of less biomass, as this favors their praise, but the ecosystems tries to balance the flows of energy and to be able to maintain more or less a sustainable balance. So we have three levels, three hierarchies with different divisions. One with populations, systems, and the other part of the scheme is when we do the resources. There is a system, social system that is they benefit from this production system. If that production system varies, well, we can see the system of governance affected, which is a system. We're going to concentrate basically on the biological part of population. Populations that are components of ecosystems to be able to carry out this part of the resilience. And this is more or less what we can identify the resilience of populations. Our population grows more or less how the graph shows it, and I'm going to put some equations that they have always told me not to, but they are very simple. I'm going to explain to you very, very shortly why. These are equations that are basically based on some subtraction, multiplication, division, and this for you to know where the indicators come from, and that you can understand that it's complex, but in reality they're not. And if our population grows very quickly when we exploit the population, the biomass decreases, and the black areas that refer to the production areas according to the level of production. There is a definition of the macro sustained, where the population is at the maximum capacity of production, which is the darkest spot in the center. And the resilience in the population when it is affected by a biomass is measured by the amount of reposition of that biomass. For example, if we see from the level A to beta, alpha to beta, in a population, the time it takes to recover that biomass has to do with the resilience. And that has to do with the rate of growth of the population. It's not the same population with a history of thousands of longevity or one with a short life. The reproductivity is lower, so the capacities for recovery or regeneration of biomass and the population that is resilience itself expressed by the amount of rate of growth. The thing when we see it from the ecosystem, the current vision in the fishery is that the fishery retains biomass in the population that is affected, and what we have in the fishery is the other populations that are directly affected by the fishing. This is a partial view of the ecosystem, and this is what this area on the right refers to. Left, sorry, refers to. And when we do the fishing, there is a recovery. Through this, we affect others, and there are things of conservation that we have to see there. The matter of the classification of the habitat, contamination of the habitat, contamination caused by the fishery. So the point is that this vision is this is related to the matter related to the objective and the one that has immediate consequences. The rest of the ecosystem we don't see it. So if we receive the ecosystem from the fishery, we would only have a partial view. The point is that from the population to the ecosystem, the point here is the other way around. Look at a holistic approach where we have the attributes of the ecosystem, where we might be able to determine how to exploit a population in a sustainable manner. A trophic or network is more or less represented as seen here on the left. We have some species that eat others, and there is, I don't know, expression of migration, etc., that had to be contemplated by the central part is the trophic relationship and the relationship of those. The scheme on the right, what it pretends to represent is precisely the trophic chain that we see on the left. When we build a trophic model, we put the biomass, what each species represents, and we see the information that they consume, the prey and the predators, and we quantify the columns. On the right, we have the prey and the predators. So we can see how much each one of the predators makes of these prey, or how many prey are taken by the predators. So this represents the flows, the position of the I represents the prey, and the position of J is the predators. The point, according to this position, would reflect the sum. So the position of the I, we have a circle with a J. This means that all the prey that consume predator J and so forth. If we have the position J, it would be all the prey. Out of all the prey, how many predators eat out of that? And it is easy to calculate if we have the information. That's a problem, of course, but this is simple sums, subtractions, multiplications, and divisions. And I put it like this because it is very interesting for me to see the detail, because it's something that is relatively complex, but it's also very easy to understand. There are some things that we want to divide the concept of how to look at the ecosystem. If we calculate the flow from one to the other, and we relate this to the amount of flows, we have the probability of having that new. But if we can measure that new flow from one to the other, we can also measure the probability that it's not going to go to that nodal, that there might be a possibility of occurrence and a possibility that this might not occur. And that possibility of occurrence would be the uncertainty. There are flows that are known, well, but the rest of the flows, it is an uncertainty in regard to the initial flow. The other point is that if we multiply the probability that are represented in that indicator, the flows of the ecosystem, the total flow of the ecosystem. Now imagine a scenario where you have the flow chart, but the probabilities are completely random, where all the individuals, all the flows are completely random with the same probability. That characteristic, measured in terms of flow, would be the entropy of the system. So entropy would not mean, it would be determined by the collectivity. But the possibility is completely random. In a system, in a real system, that is not my situation. For example, we have the famous ideological appreciation of qual consumes the eucalyptus, or we have bamboo to be able to maintain the system, the ecosystem of a panda bear. If we disappear the bamboo, the panda would disappear. So it would not be the same ecosystem because there would be something that would be missing. The other species consume bamboo, and other species they remain. So the point here is that the flows that maintain the appreciation, for example, the coral reefs, they have the topic of relationships, and that is the conditioned flows, because they cannot disappear, or if they disappear, we're talking about a completely different arrangement of the ecosystem. So we can characterize this type of information. This is what we call average information. If we have the calculation of the total of probable flows, remember that we talked about this, and we talk about the condition of flows, or conditioning flows, and this is something that we just add for all the relationships between prey and predators. We have in the first case the total flows, we have the capacity for the development of the ecosystem. In the second case, with regard to the conditioned flows, this expression reflects the balancing of the ecosystem, and the difference, it's a very important concept for us right now. The difference between those two is the entropy of flows, between the energy that is not conditioned to flow in a certain way. That entropy is precisely what reflects the resilience of the ecosystem. So a system that is undisturbed has the possibilities of the conditioned flows, and we maintain the possibilities of the non-conditioned flows. But in a disturbed system, we have the probabilities of the limited flows or conditions, but we alter the possibilities of the flow of the non-conditioned. We are talking about probabilities, not magnitude. Because if we have the pi mass, of course, we reduce the magnitude, but not the probability. In this way, the system is made, the contribution of each one of the species of the groups to the relationship of the ecosystems is maintained stable in terms of probability, but not so the non-conditioned flows. And those non-conditioned flows is what we have come to call the energy in reserve of ecosystems, and this is what formally represents their resilience. And this do not take much into account the equations. I insist I put them there so that you can bear in mind that it's easy. You have to make the calculations without much problem. If we just divide what the indicator of ascendance, which is the regression between the total capacity, we have the total capacity of the organization, and if that is something we subtract from 1 and we put it into proportions, this is what would represent the resilience level of the ecosystem. So what is this good for? Well, the total resilience of the ecosystem depends on the capacity of each one of the populations to reposition the position of biomass. That table of a chart is according to the table that we were looking at, plus the entropy of flows that is reflected by the ecosystem. What do we do then? Well, if we can characterize these quantities, and that's why I was interested in seeing this, well, we can have a calculation of the relative order of the ecosystem and the capacity of reproduction of the ecosystem. The graph on the right and the x-axis, this represents the rate of disturbance. The fishery is just the amount of harvest, which is just to capture biomass. So if we have a real modeling and we take the fishery out of the biomass and we add this to the live mass, we would have the scheme of an ecosystem that is non-disturbed. If we do this exercise of simulation, increasing the harvest, we can see how this changes, but we can still do this for different groups. So this graph represents precisely the changes in the indicator of resilience in this case, between -1, which is the entropy of the resilience, and then this changes gradually, depending on how the biomass is affected. The interesting thing is that the points of inflection that we always have within a value, a range of values. It is important the rate of harvest because we can also reflect it as mortality. Mortality by fishing, and this is something that we're going to manage with. Look at it with managing. The important thing is that we have this, and we are creating the point of inflection. And the idea is that we're not going to come to that, or we're going to come to this level, because after this we affect the ecosystem. If we do this for an ecosystem, for example, on the right, we have the North Gulf of California. Each population represents a different value represented by the psychotropic, and we have an isoline or an individual data that represents the limit of biomass that you can extract as a maximum, so as not to affect the replenishment of the system. If we convert this to the rate of harvest, and then we have easily the mortality by fishery that we can determine as the limit, and in the management we can say the limit of education of biomass in the population. So there we have the proportion of biomass. And why do I refer you to a rate of harvest? Well, this is examples of several ecosystems where we have done these type of things. Let's not go too much into it. How is this important? Well, in Mexico, there are several certified fisheries, among them the one of sardines, and we have said that sardines are the base of the trophic chain at the sea, and there is a lot of concern of how much can be fished in sardines, because if we fish too much, you're going to affect the rest of the trophic species chain and other species. In Mexico, one of the first questions that they made to certify one of these fisheries is how much biomass could be extracted of sardine after the fishing. How much biomass could be extracted after that, so that after the fishing we have enough biomass so as not to affect the rotation of the ecosystem. And precisely, we use this type of resource, like on the left, to see what is the amount of rate of harvest that we could apply, and we could say that in the case of the sardine that it cannot be exploited beyond that level. That is the amount of exploitation of biomass, so that we can maintain that sustainably within the system. And a very important thing, when we talk about the rate of harvest, we're talking about the rate of capturing biomass, because this concept is compatible with the climate change. And if the population changes or fluctuates, then what we have to determine is the proportion that can be captured independent of the amount of biomass. And so you can extract 38% as a limit if there is a very little mass, and 38% of the biomass, and if there is a lot, 61% of the biomass. So this type of idea is empathic, and this type of indicators to be able to deal with fishery, tentative fishery related to climate change. And this can be calculated for any type of activity that we want to develop, be it natural or induced by humans, that can create the loss of biomass. And why the monitoring? Well, because this avoids many situations. So, very quickly, the biomass of each one of the species that we are going to see in the production of biomass, this coefficient is equal to the natural consumption of biomass, consumption capture, and we have the other model. So we have to analyze that this model is balanced. Balanced in the sense that in the static model, all the aspects of the model are lost. So the model has to be balanced. And this system of balance that we can see the ecological, biological properties and fishery that are logic and conceptual aspects. So in the production of biomass, we know that this relationship grows, reduces according to the philanthropic. We can see that, we can observe that, and maybe identify that the system is consistently acceptable. And fishery, because the idea is that the model itself should reflect the state of the fishery and reflect the state of the advancement. So we put this in, and the model is consistent. And after this, doing a dynamic, you need to identify amounts of biomass, and you can represent periods of time when the model turns out to be to calibrated biomasses. And if we can do this, then we can help us for realistic scenarios. If we involve the indicators, as I said a while ago, for these trends, then we are on the acceptable side. And I say this, I had energy to practice what we had here, but I'm going to retain that for due to time, and I'm going to leave it here. Thank you. Thank you, Dr. Abellín. And we will continue with Dr. Ileana Abreu, associated to the Pontificia Catholic University of Chile. She is assistant director of the Millennium Studio, the Center for the Regulation of the Genome, and main researcher for the projects of research in this institution. She has more than 65 publications in which we have books, chapters of books, and publications of exceptions. So, Dr. Abreu is going to present a speech, genome, as a tool for the study of monitoring of fish, birds, and in the Austral Ocean. Hola, hello, good afternoon. Thank you very much for being here present, and thank you very much to the organization for the invitation. It is a pleasure to be here. Well, I'm going to talk about the economic situation, but don't be aware, I'm going to make it as simple as possible, and I'm going to talk a little bit about Antarctic and the Austral Sea. And I will be part of the project of long-term in Chile, collaborative, and one of them is for the Antarctic and subantarctic biodiversity. The other one is the Millennium Center for the Regulation of Genome, and the other one is the Millennium Nucleus for the integration of Patagonia. And I am interested in obtaining the birds from the deserts to the forest and sea, the vertebrates mainly. And these are some of the students that work in the lab, and they have done all the hard job. Well, our lab is very interested in diversification, specification of species, adaptation, and especially, obviously, all the things that we have contributed with the conservation of several of those ecosystems. And therefore, we are always looking at a continuum of specialization of species, from a more incipient species to the complete divergence, or looking between populations. And in the middle of all that process, well, we can also have some hybridation between species. But to study the genome, we need some take sample of animals so that we can have an idea. And for that, to go to the Antarctic, we have to go across the Bering with some waves. And there we come, and some colonies like these of the penguin, and some of them more to the south, like the Delis penguins. And there we capture the animals to take measurements and blood samples. These are yellow fins penguins in the Cape. There we went with 3 students from the lab to work, and there we collected samples of blood. And well, I was 2 seasons back, 2 summers ago, we were going to begin going to colonies that were more distant of emperor penguins, because this is something that produces platforms of ice over the sea. So we started looking at some Krugers, and we looked at some of the satellite images, and precisely those 10 colonies, the Bellinghausen Sea. And what we saw is that there is an early onset of those colonies of emperor penguins affecting there this stage of monitoring. And now we continue on monitoring with the satellite images the rest of the years. But in the beginning, the idea was just to review where precisely were the colonies, and we found the surprise. But finally, we came to colonies that were not affected, like the Sea of Weddell. You can see the adults there to the left, and I don't know if you can see it really. And we also have the 4 students of lab, our collaborators who are coming close to the colony and looking at the small penguins, and with a little more security, because in South America we had the highly pathogenic avian flu, and we also had to influence this. So we took this from the samples we took from the droplings, and we analyzed it in lab. And there we have the emperor penguins. There we capture them. We went with that large ship, which is an icebreaker. There I am on the platform of ice, getting blood samples from the emperor penguins. And also, we wanted to see how affected this was by the climate change, the early melting of the ice platforms. And here we're capturing birds that were already changing their feathers. So we are putting there the satellite transmitters that now it is being monitored, and you can see the penguins here. They're going back to their colonies with the transmitter on their back. Just like Happy Feet, you remember the movie Happy Feet, okay? And then we go to the lab in the vessel to sequence this, and then we have the molecule, and here we have in Chile, and here we have the genome, the human genome, and this is sequenced at 2 years, and this is put the genome in 1 week, or hundreds of genomes. But there we have a great amount of data that we have to work on, and a great part of the year we are entertained in terrains. So we are there connected, looking at the supercomputers, because we have a great amount of data, and working with the genome sequence. So what we do basically is look at the genomes. We assemble the genomes of each one of the individuals, and we compare between the individuals. And this that we see is this SNPs, the variation of the genome between individuals, and what we call SNPs. Then I'm going to talk about this in another presentation, which is basically the polymorphism of the unique. Okay, so. But before talking about some of the jobs, that it is important to mention that we have the characteristics that are very important of the South Sea, and these are something that is important with feeding from high temperatures at north and south. So this is something that is important for the species. Apart from that, they have determined provinces in all of the regions. It's for the biodiversity, and it's mainly for the coastal areas. And well, obviously, talking about the Antarctic and the South Sea, I have to talk about the penguins. You're going to understand this, why approximately 20 species that are distributed between cold waters of the southern hemisphere. And the first job that I'm going to show you is a comparison that is more revolutionary, a comparison between species. It's something that is a reconstruction of the genetics of all the species of penguins, and we can see some very interesting relationships. That we had doubts in regard to the position of the king and emperor penguins, and we can see that the first group is there, and we see that the group diversified in approximately 22 million years in the Neocene, and in New Zealand and Australia. So we can reconstruct the ancestry, and this is something that appeared in all the templates. All the penguins are algae, or they're kiwis, and they are noctactyl. And so we have also reconstructed in the graph on the top part the environment towards the past. In other words, the ancestry niche. And we saw the maximum temperature of increase of 9 degrees. We can also see that some of the groups, like the Victus, that have a hood, there's a high level of introduction of the evolutionary process of its species. Well, now I'm going to talk a little bit about the process, microbial evolutionary process in the species.
Penguins have two main characteristics: dispersion, philopatrics. They belong to the same colony. They don't socialize among populations. Those characteristics are contrasting. And I'll talk about some studies that we are doing with this species. The first one, Eudiptes, the yellow feathered. One of my students, sequence three superior groups, yellow feathered, and two inferior ones. Macaroni royal has a white face and yellow feather. We didn't know if it were 1, 2, or 3 species. And we found out that there are 3 species of yellow feathers, superior groups, and the northern one also marked here the separation. It's a very important barrier, it's a subtropical area. The southern Macaroni royal blue are separated as a species. We studied more in depth genomic data, more than 5,000 SNPs, and what we saw in yellow, we find the royals, white face, and one phenotype that increased their frequency. They are not separated as a species for macaroni, and we had 2 genetic groups, main genetic analysis, structure analysis, 2 groups of 2 colors. Important effect, not as a species, but as population of the polar front. We want to see this debate, how many species were there of the yellow feather. Maria Jose Furgonia studied again parts of the genome, more than 14,000 SNPs, and saw that clearly different methods to delimit the species. There are 3 species which are different: southern South America, west Indic Ocean, and the other one separated by the tropical area. These are very important separation barriers, and with these 3 elements we could establish the 3 species interpopulation separation. The northern 2 groups in green, in 2 islands, South America, in the Falkland Islands compared with the rest of the continent. In another penguins in Antarctic Peninsula, we find Adelie, Barbijo y Papuas. Adelie y Barbijos, the population is decreasing and they move behind the creel. They have low genetic mutations. But the yellow feather, as I showed, have differences as a population. It's a different species from the other, from Barbijo. they stay there in the same colony throughout the year. Every year I go to that colony, they are there. We knew then that Antarctica, we have very organized colonies. Darwin, his doctorate thesis, and some other publication, we saw that Antarctic ones are very different genetically, even to the species level, compared to the ones of South America, separated by the polar front. And we saw the groups in the Indic Ocean, Macquarie and others, Kerguelen Island in green. We saw 4 genetic groups, very well different at species level, and we saw the differences between these taxonomic groups. We analyzed parts of the genome that are which sequenced full genome, and we saw genes specific for taxic in Antarctica yellow. We have anthropogenesis, which are adapted to the cold weather in blue, genes as it makes sense of what we are founding. We evaluated the niche of 4 taxonomic groups, marine and land, using satellite data and the environment. And we could see in this sphere in blue temperature, colonize Antarctic, and we saw red, to adapt to less temperatures. On the other side, to colonize yellow, South America, adapted to higher temperatures. But developments have to do with their feeding. Another study, Lucila and Josefina, with the squaws, predators of the chicks of the penguins. You have 3 species of squaws, some subspecies, but basically 3 species: polar in red, Panta in green, in Chilena, South America. What we can see were studies that were not able to separate these 3 species. So we sequenced the genome in 20 locations, and what we saw, yes, there is a support for the 3 species with in analysis of the main component and the genome structure, separate 3 colors. The structure we can see in red square. We have a mixing area, which is interesting. In a big area, you can see some there figures where we have a mix between the 3 species. This can be a transfer of allelos. And the last example, Peniscos penguins, just to show with genomes we can estimate the effective size of historical aspects. A period that goes back to 10,000 years. These Galapagos are decreasing compared to the African. And another analysis where we can see the change in size to the present. Analysis that can be done to contribute to understand data for conservation purposes. Take-home messages. Oceanic species have an interesting effect. These three groups I mentioned, not other species of penguins, morphological differences in the case. Doesn't mean necessarily different evolutionary units or vice versa. In the Papua or the yellow feather, there's no clear identification of a species. Macaroni royal, we have genotypic but not genetic differences. Important to delimit the species, as I mentioned. The krill is not growing because of climate change, and Adeli the population is going down. Papua is expanding. It has a less concern. As we see in this figure, in red, size of population in Antarctica are high. Also in yellow from South America. As you can see, very small population sizes. We also did projections of future niches. We can see in Antarctica they increase the populational sizes. They keep growing in the future, but in other regions, like South America and Chilean Islands, population will decrease. So it's important to delimit species to have measures of conservation for each one. For example, in the case of yellow feathered, southern and northern must be considered as a different species, and the southeast, those of the east, populations are decreasing 80-90%. Lastly, I want to tell you, coordinating a project in Chile, we are beginning this year Mil genomas chiles to sequence biogenomic of the biodiversity in Chile. You can access our website. We have an important participation from the citizens, where we have a group, a taxonomic committee, go to popular vote balloting, and we're going to sequence the buru, which is a small deer. We will sequence plants also. We have a contribution in terms of genoma techniques, and we provide these tools to different schools. Thank you very much.
¿Quieres ir a estudiar pingüinos toda la vida?
Thank you, Dr. Abreu. Next, we have the participation of Dr. Andrea Garcia Rojas, researcher of our School of Biological Sciences. Dr. Garcia participated in different research projects, evaluation of health and microplastics findings. Her presentation the activities of our research school in biomonitoring. Thank you. Thank you for the students and colleagues that are still present. I thought I would be alone this afternoon. I have the mission of telling you what we have been doing for 50 years. The school began in 1973 together with the National University. When the university was founded, we had a developed process to what we have the School of Biological Sciences at research level. During the 70s to date, we began with two big lines, the biofisheries and aquaculture. As time went by today, we can talk that we are still dealing with these two lines, but we also are studying ecosystems like mangroves and phytoplankton, marine microbiology, microplastics. So I'll begin to tell you about, as academic unit, we are contributing to the monitoring of coastal marine environment of our country. In fisheries, monitoring for many years has been developed, as we know now, as the lab in Punta Arenas area. We have several lines of monitoring, for example, feeding preference of different species of Golfo Nicoya, mating periods, species, areas, growing areas very related to species of commercial use, identification of the physical-chemical influence, dynamic in populations, evaluation of species with economic potential. All of these have been part and goes together with the fishing in communities, artisan fishermen. We work very close with these communities. Today, when we were beginning this symposium, my friend Luis Adrian Hernandez was doing a presentation about the contribution of the evaluation of the artisan fleet. Another topic we have been developing in science biological lab is apiculture. We have to underline that monitoring related to what we know as apiculture is something that comes from one of the most important manifestations that we have to have as a university, which is the tech preference. The part of apiculture with the program regionalization of the National University, we've been able to accomplish the implementation of oyster farms in one of the main Gulf area, Nicoya. Through this tech transfer to these fishing communities, we must have an accompaniment together to monitor physical-chemical parameters, microbiology related to the Gulf of Nicoya and these farms. Together with them, we have a program, multidisciplinary program, ECOMAR, where the four universities, University of Costa Rica, TEC, and UNA, and UTN, are participating, doing an evaluation assessment of emerging polluters, so to provide an added value to those products, those oysters, to have a green label, green seal, free of emerging contaminants. The work we do, you can access more information in this panel in Thermic Dome Hall, small scale apiculture, which is a fantastic work that began here in National University. Also, as part of the monitoring we do in the lab, phytoplankton is one of those monitorings we've been doing since the 80s. in our coastal areas. We have been monitoring phytoplankton and algae diseases. Our lab has become the reference lab to provide follow-up to red problems. Red tide. Something that I always want to stress is that the work that goes together with apiculture is the collaboration in the analysis of samples of phytoplankton for the care of oyster products. This is an accompaniment provided to coastal communities, and I stress this about many of the projects and the labs we have in biological sciences. We work together very close with the communities. Also, you can have more information on Wednesday at 9 o'clock, where about 2 decades of monitoring in the Pacific area of Costa Rica. Another big programs of monitoring, mangroves. Mangroves began its monitoring about the 80s. The target was to see the structure, organisms, mollusks related, and after the 2000 year we began a work where we were able to generate a national protocol, a protocol that should be used in mangroves of our country for its monitoring. And the origin is in the National University, and with a collaboration with the National System of Conservation, SINAC, 26 indicators that allow to take a decision about what is going on in our mangroves. As you can see in these pictures, with SINAC and university are monitoring in situ. This project have had some results to have permanent parcels in 5 mangroves related, 5 areas in the 10 Pisca areas to estimate indicators of structure, coverage, granulometric, heavy metals. This is another one of the big jobs where we began collaborations, multidisciplinary, for executing monitoring. And we have another one of big contributions that we do from the unit, the academic unit from biological, which is rocky reefs. Began in 2018 with a vision. We wanted to determine, according to biodiversity of protected marine areas, if the measures were effective in terms of protecting the area. With this image, in one marine area protected, which was successful, we can have an inverted pyramid where we can have big organisms that would allow me to have control other levels. In conclusion, we could say that areas that are not being protected, this pyramid would be a normal one, but we could have a dominance of echinoderms. When we began this monitoring, my working group established National Protocol of Ecological monitoring of rocky reefs implemented after 2021, and is based in a monitoring of subaquatic transectors, where we are evaluating all the vertebrae and the ones we all see in transector. It's a work we do 3 times at 2 depths, and this protocol is related to around 18 indicators with which we normally are going to try to measure all the organisms into evidence, to densities, coverage, biomass. If one protected area or an area non-protected, if they're healthy or not. In this figure, one of the analyses we have of the first applications of this protocol, and we did it in some protected areas of the North Pacific and Cocos Island, as we expected. The first point is Cocos Islands being an area so preserved with so many measures in place. Is a marine protected area which is really healthy. But we have some other areas that don't have so many possibilities where we can find the evidence in the results. Another fundamental part of the doing of our lab is microbiology monitoring, abundance and biodiversity of luminescent organisms to determine those microbian communities in the Gulf of Nicoya from the work of Carolina Marin, biotechnological applications of organisms from these areas, and we have a long way to go in these themes. So this is advances that evidence how from the school we are working from the quality of water, microorganisms to superior organisms and ecosystems as such. Another line that we have been developing from the microbiology lab and through coastal marine system is microplastic monitoring. It has been based in sediments and organisms, marine organisms tissue. We have been working from 2018 in Baulas Park, Cocos Island, and this work we had worked with external entities and other academic units. We had projects, for example, from Cocos Island that is now in a third stage with collaboration of FICO. We did a study of the 2 most important Gulf, Nicoya, Golfo Dulce, interdisciplinary work with a FES project with the people of the University of Costa Rica and the Technological Institute. And we have the last 2 projects. One that was to evidence the microplastic, not just a marine problem, is also happening on land, because we determined the presence of microplastic in terrestrial organisms and canned products. We have initiated a project in Cahuita National Park where we will try to determine how the presence of microplastics has related with coastal erosion and about currents. A joint work with the Department of Physics, Geography School, Photography School, and Social Planning School. What we have found, what have we found in all of this monitoring? More than 60% of events in marine organisms, 100% in microplastics, in sediments,
And in almost all the cases, we have a prevalence of fiber-type microplastics. And to finish, to wrap up, another one of the great contributions that are happening, and this is a very interesting incorporation because this is a colleague from biotechnology. It's the monitoring of marine fungi. This project comes about from monitoring in mangroves. So with this project, we are trying to potentiate a little bit based on those fungi, the potentiation of antibiotic-resistant agents, the potential for production of biosurfactants, and the detection of potential biosynthetic through computational approaches and experimental validation. So as you can see, the approach of the biological monitoring that we do in the school has many different angles to it, but always in favor of looking to see how the ecosystems are, to be able to also make a collaboration with communities. And this is also based on the fact that as part of the National University, we are a humanistic university, and we have to try to have our science go beyond simply saying that we're doing science. This science has to be transferred, it has to be taken to the community as a whole. And that would be all on my part, so thank you very much. Thank you very much, Andrea, for the summary of what our school does. It's an honor to be part of this National University. We are going to open the floor now for questions. If anybody wants to make a question, pose a question to the panel, please raise your hand and we will have the microphone come to you. Thank you. Sorry. Well, my question is very general and might be maybe even somewhat difficult to answer because I have still not found somebody to be able to give me a certain answer because I know that it's difficult. And it was because it was mentioned by basically 4 of the expositions is, in what way can you make you as current researchers and we as future researchers, how can we make our studies and our findings, our science, reach and merge with the politics, because the politics and the science should be basically best friends, and they are not. And that has been a big problem to us because we end up, the people who end up executing everything as the politicians, the social part. So I don't understand in what way you as researchers and we as future researchers, how can we have this executed in the way that we see it? I know that it's complicated, but I would like to maybe be guided a little bit as to how to make that. How can we reach that part? If somebody could give me that answer, I'd appreciate it. Well, our first step would be the participation in those spaces that are opened up by politics. I think that sometimes we the scientists don't want to do so. Okay, we are having a new law enacted, and we have to present our opinion in regard to that to see if that law has something related to our environment of action. So I think that we have to start there. And in that participation is when we can go and exhibit something. And we can see how in the generation of information that it should be easy to understand. Why? Because the scientific communication is something that not necessarily the politician is going to understand, so there has to be a transformation. We have to be able to disseminate beyond science so that that science might reach. The opposition talks about microplastic and things like that, or topics that are very high related to genomes, for example. The scientist is not going to understand. We have to transform that communication, that scientific communication, turn it into something the public so that it can reach the politicians. In this case, the politicians. I completely agree with what you have just said, but I would also point out that it is not our responsibility to do so. In other words, if the politicians ignore the science, the way it is happening in many places around us, we simply have to stay firm on the fact that the science says what it says. But it's not our job either to educate the scientists, the politicians, sorry. Our job is to educate the general public, but not the politicians. And maybe what happens is that some scientists should become politicians, and that is something that to many of us is very difficult because it is a change. It represents a change of language that is not based on evidence. It's based on the propaganda. It is something that is very difficult to accept, but that's something that we have to deal with, to think about it. And another thing that is very important that I think, you have to make your science become important. And in this sense, you have the help of the learning. Because when you do science that comes out in the big, the larger communication media, and when you do a job that you know that it's going to be on a very important interview, that this job answers a concrete question, the media is going to pick it up. Because they like it, the media likes it. And so the politicians have more difficulty ignoring you, because you are scientists at an international level. It's difficult, but try to do it every day more. Meanwhile, just to complement something, I agree completely, and also all the scientists have to make that task. All the universities and research institutes should have scientists that dedicate also to that, to elevate to public policies the scientific knowledge that is generated, for example, in a university or an institution. Why? Because many of us have many other tasks. And we want to work in the lab, we have to obtain funds, we have to teach, and so many times not everything has to be, everybody has to be dedicated to that, but we do have to present an opinion when they are talking about our area of relevance, or to see if we have magazines at the national level that are going to publish this to the general audience and the politicians that are going to reach them. Okay, so what is the recipe? The recipe, what is it that we do in Mexico? Well, at least there in the what the UNAM has done in Yucatan. It is to look for these allies, to look for allies. And one of the main allies that we have are the foundations, and the other one is the organizations, the NGOs, right? So they are in charge of having their legal bodies, their legal advisors, and their groups for dissemination of the information, which are the influencers that they have, who manage all the information. So we do that union for the solution of a problem, a specific problem, and we generate as a university the hard data that they require. And each one of these parts, both the university and foundation, as well as general society in general, is in charge of implementing at the different scenarios in which they have to be. And with all this, we come to generate public policy. It's not immediate. It's not something that we can do overnight. For example, our law the official norms have been improved thanks to this joint effort that we have done. Well, I'm going to differ a little bit, especially with my friend Pepe and with my co-patriot. I'm going to differ because I believe that it is responsibility of the scientists to come close to the politicians and to establish a dialogue with them. For me, we have to create formal structures that will facilitate that communication. Formal mechanisms, legal mechanisms that will facilitate the communication between the scientific environment and the political. That communication, apart from that, also has to be done at several levels to create a structure in which we might have maybe our Minister of Science sitting with a Minister of the Environment and Minister of the Agriculture, but also that communication has to take place at a technical level. Because if it only happens at one of the levels, it's not going to work. There has to be this communication. And that communication is only effective if you generate confidence. And if you can generate confidence or trust, you have to spend time and dedication. And what cannot be done is what I have seen many times from the scientific environment, is to come out in the newspaper and go against the scientists and wait later for them to come and sit down at a table and listen to you and do what you say has to be done. I understand that there are some politicians with which maybe there is not a way of sitting down ever at a table with them, but there are others that might listen to you. But they will not listen maybe the first time, or they will not listen and understand the first time that you sit down and talk to them. But if you work on that, and I think in the center where we work, we have a very good relationship with determined ministries, and when one of us sits at a table and gives an opinion, we can hear that opinion with a lot of respect. And that is something that is not built overnight. It's not something of just having the scientific evidence at hand, but also to convince and to see that the person that is listening to you, not necessarily to know all the scientific process behind it, or all this person trusts them, and I think that is something that can be built. It's not our responsibility. We cannot leave it in their hands. That there are many ways of doing it, but we cannot leave it up to the influencers. So it's something we have to do. I don't know if I'm going to disagree with Giuseppe, but that's not the point. It's something that we have to understand very clearly, is that normally in the research and the academic sector, we're not responsible for the decision making. To be interactive, we have to interact with the people who make the decisions. So, as I was telling you, how do we comment this? Of course, there is part of the science in which you cannot apply this immediately, but there's another part in which you can. One of the essential aspects, is that we also, as researchers, linked to the seriousness, we have to identify problems according to the decision making. So that after that we can propose not hypotheses but questions for research. The questions for research are normally aimed at responding a particular problem. An example, we give doctors and PhDs and master's degrees, and normally in the case of the fishery we don't ask for hypotheses. We ask them for a scientific question that has to be answered. Why? Because the evaluation of stocks, there is a hypothesis, but it generates the idea that it's for the management. If we are able to identify problems and specific questions, then our results can be channelized. There are entities that, although it seems not, but there are NGOs and international entities that are identifying what you do. And all of a sudden you have problems that you have identified, and maybe you're not able to interact immediately with the sector, and suddenly it seems that it does. And they're the ones who are going to consult it. What we cannot lose is that channel of communication. To be permanently linked to the people that have to make decisions. In Mexico, for example, in the case of fishery, there is an Institute of Fishery that is responsible for giving the rulings that the National Fishing Commission is going to generate so that to establish the rules that have to be followed. We're going to deal with this because the Fishing Commission Commission is the one that determines the conditions. But the Institute is the one that carries out the research. We do not make decisions, but we can work with them, inasmuch as it is common, the identification of the problems. And there we have the FAO, and we have a series of things where the certification, for example, is applied knowledge. I don't want to mention this too much, but what I was asking just a little while ago, how much biomass do we have? And this concept, it was generated by a question that was posed in a certification process. And from that, we have developed a series of things of how to use it. And then we have other things, and we continue on asking, but it is that vehicle, it's a vehicle that is used. Thank you very much for the question. Any other question? Okay, once again, I think that I do have a question. But to begin with, I would like to congratulate the level of this panel. I think it seems to me incredible. It seems to me a very good conclusion from all the different parts of the world. And this leads me to my second comment. And it's good to hear that Andrea and Luis are in this panel. And as a graduate from the UNA, obviously this is something that makes me very happy. With regard to this topic that you're talking about in the consultation that was made by the student, there is an undoubtful triangulation that I've been talking about for a while now, and that we have it in the academic sector. Sometimes they don't understand me very well, which is the triangle, society, academy, and institutions. A clear example of this triangulation, we could say that it is part of the job that is done by the National University, by saying that we work in protected areas, that it has done research in different parts, and that we can compare how the results of one area that is completely protected, like Isla Coco, has better health than one that, although it is protected, we have an anthropic situation. But in that process, it is being part of the institution, which is the entity in charge of the preservation of the resources. And well, at this moment, I am at pause, but I also belong to the institutionality, to the National System for Conservation Areas, or areas of conservation. And from that foxhole, it's sometimes difficult to get close to the communities because there is a lot of shortage of resources. And on the other hand, the communities also sometimes don't want to be a part of it, because many times, unfortunately, there's very little information, as I mentioned before. And also, the people who are the people who like to get involved in everything, not necessarily do they have the leadership, but they have some interest, and that is why from the institution we have to do it, and from the academia we have to filter who has a real interest and who doesn't. So my question is, before the question is, I do believe that the entities that carry out research have a very important part in educating the politicians, because unfortunately, not all the politicians are brilliant. Sorry for saying that like that, but this demonstrates the task to communicate that information. Regarding the example that was being mentioned by the doctor of the Institute, Giuseppe is your name? Yes, gentleman? Yes, okay. The other Spaniards. It seems to me that it is a very solid structure. How were you able to acquire, and this is my question, how were you able to be heard? Because it gives me the impression that you are a very important part of the decision making, although you are not a governmental entity. To make that a link with the government and to be heard, because this is a comment that I believe that in Costa Rica there is not a lot of seriousness, or at least things are not taken seriously. The institutions that contribute to this type with this type of information, or they are the same type from the governmental side, like the topic of Incopesca. And how have you been able to achieve it? And do we have the right impression that you are well put together and that you are being heard? Well, I am older, but maybe not that much, because this is something that started many years before me. But I believe that it is a problem. One of the advantages that the institution that I belong to has, and I remember that in view of the explanation that was given by the previous director, he said a thing, and he's right in saying that when the Spanish Institute receives a question or a requirement of advisory, that requirement comes to the director. That director speaks with the assistant directors that he has underneath, and that falls until it comes to the community of scientists, and we create a working group to resolve that problem, and that report goes up and it is signed by the director then. So that what it has to do is to filter that type of interested comments, or sometimes maybe we might be even accused of guiding politically so as not to hurt with a question. So I believe that there always has to be a balance there, saying that we are humans and so forth, but at the end it is the credibility of the institution that is at hand here. The question is not to an institution, to a researcher, but it is a question to the institution. The one who signs it is the director. This has its disadvantages because there is it subtracts from the credibility that the person that is responding to the question has. So I guess that any question, any group, for many years, there are moments in which that communication works better, or sometimes it works worse. And we have to try to do this. But I think that the credibility and to generate that confidence is something that is fundamental. All the structures are created as this happens. And also to expand what we said before, in theory, the politicians, the level of the technicians at the ministries, at entities of this level are much more prepared than the politicians. Those are the ones that we can talk to. It's the politicians that I can believe that this is something vain, because apart from that, the politicians only last 4 days in office, let's say, luckily. And that is something that we have to say. I think that the institute that is presented here, they have worked a long time with this intermediate level, that are also technicians, that at the level of the institution that they have to solve problems. We have to tell them solve this, and therefore somebody has to say, well, we have to make a report, we have to think it over, we have to measure this, we have to measure that other thing, and well, but I need a response for it the day after tomorrow. Why? Because it was asked by a politician who doesn't know how to wait. Yes, I agree with you, but the thing is that the politician that is there at the end depends on the technicians that he has below. There is no reason, but there is nothing else but to depend on them. That he depends on them. But at the end, remember that you have a relationship with those intermediate levels that have been very well established. But at the end, it's going to end up evolving with what we have on the top. We have some cases like that. And I would like to make a comment, a very particular comment, of what I understood about the interaction with the communities. A while ago, I was saying that we have to identify the problems and to make particular And this is something that has to be included with the communities also. If there is a community of fishermen that have a common problem, and we can identify the question, well, they are the ones who are going to help us solve. That part of the monitoring, that's something that contributes to this. This is the part where they have to answer a question that is sometimes convenient to them. So we have to identify those problems and those particular questions that are required by the communities. And it's something that can be done differently. Now, sometimes you just come to a conversational, and from that something comes up, and there are things that are very important because in the fishery part, it's not only the resources, it's also the people, the management of the resources. The management of the resources is for the people, and the fisherman understands that. And so many times, fishermen, if you speak with them and explain it to them correctly, they accept the regulations that are established for the resources. But there are other aspects, the socio-economic aspect, that you, as a researcher, you have to understand that you're working to generate knowledge for the management of the resources. That's for the people. And the problem of the people, what they see is this: the situation of poverty, the situation of social liberty or freedom. And I commonly make a question to my students in the sense that if the sustainability is the objective of the fishery resource, and I tell them that it's not. And this is perceived as a stability. But whoever is poor is going to end up poor, and whoever is rich is going to continue on being rich. And the question is sustainability and development. So we have to understand the topic of management, not only in the biological of the biomass that is available for the fishing, but also to contribute with the estimate of indicators associated to the well-being, social well-being, poverty, food safety, and even health. If we are able to identify all these type of parameters, we can project some sort of scenario where we might be able to favor the fishing sector in this. So, but for this, I insist, we have to communicate with people, and the people are going to look you up for this. Good afternoon. First of all, I would like to thank you for your time, your contribution to science, and for having shared that with us. I want to continue on with the topic that we have been dealing with, that was mentioned by the colleague Ginger also. And this also has to do with part of my personal appreciation. I consider that at least there can be conservation without involving the communities. And in this sense, for example, in Costa Rica, we have a particularity with a perspective, and it is that we do develop by public policies, and we have management plans and so forth. And in some courses, we've been told that we have excess of that, but that they are not complied. And in this sense, what I would like to point out is that, okay, we need to come to the people at the different institutions, but it's important for the population to agree that it is necessary to advance on this line of generating laws for the protection of the oceans, in this case. You as researchers, people dedicated to science, you contribute with a lot of knowledge. And the thing is that we already have a lot of data that is very alarming, but the people are not alarming. The people really is not aware that we continue on generating data that is very alarming, and they are not accepting this, so they're not interested in it. So part of the competence that we have to do is that we have to make these data reach the community, and the community has to be interested. And so my question is, from your investigations or research, what strategies do you use, in case you have them, to be able to communicate those results and involve the communities? For example, I know that in the fishery sector, it is almost mandatory to work with the fisheries community, because at the end of the day it's them, and therefore the coastal communities, but also the topic of microplastics, in the sense that the microorganisms that indicate the health of the ecosystem and how we are affecting that. What do we do, or what do you do to be able to reach the people and to be able to extend this beyond them? Because if we just continue on doing science, we continue on reporting, but in reality, the people, in a certain way, they are still not reacting.